So we might want to group our cells into clusters. And before even starting to do the clustering, we really need to consider what it is we want to cluster. So when we talk about cell types or cell identity is probably a better word because that is a bit more broad. It could be anything from the actual type of cell, how it's reacting to different environmental stimuli, where it is in development, where it is in cell cycle phase, or where it is in the spatial context. So cell identity can be many things, and you need to consider really what it is you're looking for when you're clustering data. So yes, as I said, it can be anything that performs a function or has a specific location. We can talk about both cell types and then subpopulations of cell types, and then cell types that have a specific state. So I will try to go through a bit of basic clustering theory, especially a bit of graph theory, and then talk about different distance metrics we can use, and then show a bit of single cell RNA-seq clustering and examples of other tools. So what you have now is that you measured gene expression for a bunch of cells, and we want to group them into some groups that make sense, and then say that this is cluster oligo, this is cluster neuro, and so on. But already when you start with your experiments, you have some sort of hypothesis. You think about what are the cell types? How many cell types do I expect in my tissue? How many clusters do I expect in my tissues? And you make a lot of choices based on whatever hypothesis you have to start with. So you select different number of genes depending on how complex your tissue is. We talked about this in the earlier lectures. We also define similarity in PCA space, perhaps, is the most common, and then how many components do you include? And then there's a lot of hyperparameters of these different methods that we can tweak. And different choices might lead to different results. So it's always good to keep an open mind, try a few different things. But if we get really technical, what is clustering? So the definition I would say is we try to organize objects so that everything that's more similar is grouped together and stuff that is dissimilar is separated. And some classical methods, hierarchical clustering, k-means clustering, I will go through briefly just to explain what they are, and then talk a bit more about graph-based clustering. That's the main method that's being used nowadays in single cell data. So we will have variation within a cluster. Oh, sorry, I always click here. And we will have the variation between cluster and in the whole data set. And the whole process of clustering is that we're trying to make the within variation as small as possible and the between variation as large as possible. And there are different ways then to optimize this function. That's what we're trying to achieve. And the most classical way is to do hierarchical clustering. So we define some sort of distance between all our data points, and then we build a tree with this data, either top down or bottom up, so agglomerative or divisive. And then we will have this dendrogram or tree that shows where we did this merging. So 3 and 6 are similar, they are grouped together. 2 and 5 are similar, they are grouped together. And then in the next step, all these 4 are grouped together with this sample 4, and then finally sample 1. And then when we want to separate this tree into clusters, it's very simple because we do a graph we do a cut in this tree. So if we do the cut here, we'll have this cluster with one sample and this cluster with the rest. Or if we do the cut here, we'll end up with 4 distinct clusters. And that's pretty much what hierarchical clustering is. It's simple, but you can make it quite complex because the biggest problem is how do we define distances, not between single samples. We start with defining pairwise distances between cells, but then when we start grouping them into clusters, we have to define distances between groups of samples. And the classical ways is single or complete linkage, just the closest 2 points in space, or the most distance, or the average of all points in 2 groups. Or the centroids, the distance between the centroids, or this a bit more complicated where we're trying to minimize the square error when we merge 2 clusters. So using all these different metrics of distances between groups, we can get quite different dendrograms and quite different clusters. Another typical methods is k-means clustering. The thing is here we have to define beforehand how many clusters we have. So we randomly select 3, in this case, 3 points in space as the cluster centroids. We then check which samples are more close to each of these centroids. We reassign those to those groups. Then we recalculate the centroids as the middle of these groups that we calculated. And then we iterate over this either for a certain number of iterations or until no more changes occurs. And here, just as with H-class, we can use the same sort of similarity measures between groups of samples. So these are classical clustering that works really well when you have less complex data, I would say. With single cell, it's hard to find one distance measure that capture all the complexity you have if you have too many distinct cell types. If you have a few cell types, it works really well just to do one of these classical methods. But what is super popular nowadays is to use graph-based clustering instead. And to explain to you what a graph is, I took this nice illustration that's called the Graph of Thrones. So it's basically all the characters from Game of Thrones and how often they interact with each other in the books, or actually how often each person speaks to the other person, I think. So some definitions here. Each person, in this case, is a node or a vertex. Each link between 2 people is an edge. In this case, this graph is weighted. So we have thicker and thinner edges. So they have different scores. And in this case, we also have arrows on them, so they are directed. Each node will have a number of edges, and that is called the connectivity. We will also have some nodes that are more central in the graph, and those we would call hubs. And now in graph theory, they don't use the word cluster, they instead use the word community, but it's more or less the same thing. We're trying to find groups of nodes that are more similar to each other than to the rest of the graph. So the advantage with using graphs instead of this classical method is that it can capture the multidimensional features of single cell data better. So it is quite clear that we can get nicer clusters that capture biology better also with these kind of methods. So when we have graphs, the main graphs you will see for single cell data are either KNN graphs or SNN graphs. So KNN graph is basically the K nearest neighbors. So if we take the 5 nearest neighbors of every cell and just draw lines between them, we have a KNN graph. Then we can take the KNN graph and recompute it into an SNN graph. So basically calculating how many shared neighbors each cell or node has in the graph will create an SNN graph. And it's a quite common measure of shared neighbors is this Jaccard index. So the shared neighbors divided by the total neighbors they have together. But there are other options on how to score this as a graph, like shared rank number, just the number of shared neighbors, some overlap coefficients. And then in some of these methods, we will also do pruning of the graph, which means that we cut some edges. So I will show you here a bit more clear exactly how it works. If we have a very simple KNN graph with these 5 nodes, we calculate the shared neighbors. So C and E, for instance, only has D as the shared neighbor, so that it has number 1. While C and D has 3 shared neighbors. It's basically all the other 3 groups. So then if we want to calculate the Jaccard index, it's actually 3 of the 3 other neighbors that are shared here. So it has a Jaccard index of 1. It's sharing all its neighbors. Or here it would be 2 shared neighbors, but the total neighbors is actually all the other 3. And then if you want to do pruning, it would basically just be a cutoff on this Jaccard index score. So these edges might be cut off if we set the cutoff above 1/3. So that's how the graphs are created. The next thing is that we want to put them into these packages like iGraph. And then we can either have, if we have an unweighted matrix, just an adjacency matrix. It's just zeros and ones, where we'd say that A and B should have a link. A and C should have a link, but not A and D. Or if it's a weighted graph, it's instead numbers that tell the weight of how strong that we think this sort of similarity is. And as I said, clusters are called communities in graph theory. And it's basically just having higher probability of within connections than connections to other parts of the whole graph we're looking at. That's exactly what I said. So to find these communities, we need to cut edges. So we cut it into subgraphs, and we calculate the cost of cutting. Yes, I have a question.
Yes, I'm sorry. I just have a question regarding the previous slides on the weighted matrix. So as far as I know, when I do the Jaccard index, for example, for the bulk RNA-seq, so it takes into consideration how many shared genes between the nodes are, and that's how it calculates the weight, right?
Yes, like in this example.
Yes. Okay. So now how is this applied in single cell right now to define?
Okay, so exactly what you do. I will come back, but maybe it's easier when I'm showing this. So for instance, we take PCA space, and we do then Euclidean distance in PCA space is whatever the distance measure we decide to use. For each cell, we find, if K is 5, we find the 5 closest neighbor to that cell. And then we do that for every cell. So we end up drawing these lines between cells that are within the 5 closest neighbors. Then in the next step, we calculate how many shared neighbors it has and calculate the Jaccard index. So that will be the weight instead in the shared nearest neighbor.
Okay.
So that will be the weighted nearest network that we then do the community detection on. And many of these algorithms also include a pruning step where we then also remove edges that are too weak in the SNN.
Okay, okay, now I got it. Yes, thank you.
Yeah. Yes, we need to cut the graph. And if we find the cost of cutting an edge is basically the weight of that edge. And often the smallest cut will give a lot of disjoint. It's easiest to just cut at the edges and get singular clusters. So that might not really be so informative. So instead, people come up with this idea of a normalized cut. So the whole idea is that the cost of the cut is then, in this case, 0.1 plus 0.2, it's 0.3. But we normalize it by the volume on this side and the volume on this side. So 0.3 plus all the edges out here or all the edges on the other side. And the normalized cut would be 0.3 divided by 2.5 twice. So if we instead would do this cut, where we're cutting these 4 edges, the cut cost would end up being much higher, which of course makes a lot of sense, but this is more formalized. So if we then want to partition our graph and find the best normalized cut in the graph, just with this tiny example, we have all these different ways of cutting the graph. And if we then start with a million single cells, you can imagine that this is near impossible to calculate with computers. So we need to have some sort of smart way of optimizing this function. And there are a lot of different functions for optimizing community detection. I don't understand them myself exactly, and I'm not going to even try to explain it. But here are some, and by just trial and error, I think the community has sort of stopped at Louvain and Leiden giving reasonable clusters that makes biological sense. I haven't really seen any strong argument for why those should be better for single cell than more observational. So if you want to learn to work more with networks and graphs and building on yourself, I have some links here. We have some old tutorial where you can basically take your single cell data and create your own graphs. Everything builds on this iGraph package. So actually, everything that's done in Serato or Scampi still builds on this iGraph package in the background. So if you really like to play with graphs, you should learn to use it. So now we come to the practical parts of single cell data. Quite often we start with correlations, Euclidean distances in PCA space or Euclidean distances in the integrated space that we created, like harmony space, CCA space, whatever you have created. Then the first step is building in the KNN graph with all the cells as vertices. Then, of course, K is quite important for the whole structure of this network. Then we will create an SNN from the KNN. And we'll use some sort of community detection method to find our clusters. And of course, they can also be used, these graphs can be used for trajectory analysis and other kinds of things. But for now, we're talking about clustering. And as I said, Louvain and Leiden are the most used ones for single cell RNA-seq. One issue with these sort of methods for people that come from more traditional clustering is that you can't say beforehand how many clusters you want. This is quite frustrating at times. So we have this resolution parameter that says something about how we should weight the within and between cluster sort of similarities in different ways in these algorithms. And depending on how we tweak the resolution, we will get different number of clusters. Also, if we tweak K, the number of clusters, or the pruning parameters and so on, we might get more or less clusters from our graph. What is completely hidden in these algorithms or the way they're implemented is that you will quite often have singletons, so cells that are completely disjoint from the graph, especially after pruning in Seurat, and they will just be pushed into the closest cluster, whatever cluster they're most similar to. There is an option to instead remove them, but that's not the default. And when it comes to this with distances, it's again, what gene set should we use? What space should we use? We could, in principle, do clustering on a UMAP or t-SNE, but probably it's better to do it in PCA space or whatever integrated space you have. How many dimensions should we include? And the most common is to just use the Euclidean distance in that space, but there are a few of these other metrics that are being used, but I don't think they are performing that much better. And yeah, that's exactly what I said. And of course, there's again selecting principal components that are making sense to include in when you calculate this distance metrics. Nikolai talked quite a lot about this, so I will skip over it and go to the more practical details. So in Seurat, we have first the function find neighbors. So it creates the KNN graph and the SNN graph. And the main parameters is the reduction to use, which is PCA, the number of dimensions in the or principal components defaults to 10, K, so the numbers of neighbors, and the pruning cutoff. There are other options that you can look into, but I think these are the main ones. After creating this SNN, the second step is the find clusters function. And these are the different optimization algorithms that are implemented. It's mainly the resolution we can tweak the number of clusters with and the algorithm. But if whatever you're getting here is not looking good, you sort of need to go back to the graph, reconstruct it with more or less with bigger or lower K, or even different number of principal components and so on. That's also a way to tweak what you end up with in the clustering. When it comes to SCRAN, there's either there's these separate functions for the build KNN and build SNN graph. They implement more different similarity measures, not only Jacquard, but also just number of shared or the rank of the shared neighbors. And it has a function called cluster cells that then calls the KNN and SNN and graph building. The default here for K is 10. And the default cluster function, I think, is Leiden. I forgot to write that down. And in ScanPy, it's actually a bit different because it's not doing an SNN graph. It directly just creates a KNN graph and then runs these Leiden or Levan algorithms on the graph. And the default is to have a KNN with 15 neighbors. So some comments here is that you should not cluster on the UMAP or any sort of 2D space if you have complex data. So the more dimensions you include, the more chance to detect more cell types, but also risk of including more noise and more computational burden. So as we talked about before, for like variable gene selection and so on, super heterogeneous data set with many cell types, more genes, more dimensions. If you have a single or few cell types, fewer genes, fewer dimensions, that's sort of the way you can think about it. This is something that we often get a question on, like you do your clustering, you do your UMAP, and then you have your cells that belong to one cluster, but they're not completely together in the UMAP. Some are like on the other side of the UMAP. Even though we do the same number of principal components for the same PCA, and they are still not the same. And you need to think about it, that it's different algorithms in a UMAP, or if we're talking t-SNE, it's the same. We have an algorithm that tries to optimize your data down to 2 dimensions. And always when you push something from a multi-dimensional space down to 2-dimensional space, you sort of have to make some compromise with what variation in your data you allow to be seen in 2D. While when we're doing the clustering, we're optimizing the communities in a graph with a different algorithm. They might have different choices in how it defines your clusters. And actually, if we build the UMAP on the same graph as the clustering, then they often agree more, the clustering and the UMAP, but that's also in a way cheating. I think it's a very good thing to look at these cells that differ between UMAP and clustering, because you can often see that these are problematic cells. Doublets tend to sort of shift between, like maybe they're in one cluster, but in the UMAP they're together with the other cell type. So that could be an indication of a doublet, or just low quality cells really jumping between places in the UMAP versus clustering. I just should mention quickly, I mean, you have these clustering and dimensionality reduction and so on in the Looper browser from 10x. It's running this graph-based clustering, and it's a really good first step to look at this data before you start your analysis, but it's really just run with default parameters without any quality control. So it's probably a good idea to do your own clustering also. Something I just want to mention, but that's rarely used in the single cell field, but it's like almost compulsory in classical clustering, is to do bootstrapping of your clusters. So basically, that would give you a score on how confident you are in each cluster. If you split your data and then you calculate your clusters or communities, and if you consistently get the same cells together in the same cluster, regardless of which iteration of your bootstrap, you can be more confident that it's a consistent clustering. And I don't know why the single cell community has chosen not to do bootstrap. There are a few papers where they do. Maybe it's mainly because we have a lot of cells and it's compute heavy. So then the question that everyone asks is, which clustering method is best? And as always, like Jennifer said before, you have to explore and try. It always depends on the data. There's not one single answer. Consistency between several methods is a good idea. So if you try very distinct methods and they give the same results, that would give me a lot of confidence. To do bootstrapping is, of course, a good idea, but it's also, unfortunately, the case that consistently wrong clustering can also occur, that it's consistent, but it's not biologically relevant. But in most cases, clearly distinct cell types will cluster together regardless of what you do with the data. It's more the within cell type or very similar cells that you might have an issue. So there have been a few benchmarks of different clustering methods. I'm actually jumping back to a very old comparison here. That was sort of before everyone came to the conclusion that we should do graph clustering. So they basically have here the true clusters with their different methods versus the clusters they end up with all these different methods. And in this case, what they call Seurat is this graph clustering with, I think it was Louvain algorithm, just based on PCA, which is now the standard in Seurat. So there was a lot of papers with fancy single cell clustering methods that are not being used so much anymore, from what I can see. I just want to mention also this package, Clustery, that you will see tomorrow when you do the clustering tutorial in the R exercises. It's one way to visualize your clustering. So basically, we'll take whatever metadata column you have with different values and plot a tree for you. So in this case, at different levels of number of clusters, in this case, you can see how many cells are going from one cluster to the next. And it's not this perfect split like you would have with hierarchical clustering, especially when you do this graph-based. So for instance, cluster 4, the cells that are in cluster 4 at this level end up either in cluster 3 or 5, together with cells from this cluster. So it's a good way to see what happens with your cells as you go down in resolution in your clustering. That can be a bit of a help to look at like when this graph completely breaks up and cells are just moving back and forth. It's not a good idea to cluster more and split it into more and more small clusters. Something that you can look at is, of course, differential expression. Once you have your cluster, you start comparing like pairwise between your clusters. Do I really have any genes that separate these that make biological sense? It's not an easy thing to say how many clusters you have in one data set. It takes a bit of back and forth of looking at gene signals and figuring out what you actually have in your data. There are some tools that are trying to automatically predict the number of clusters, but I wouldn't trust them so much because it can be more complex than just finding automatic groups. And always look also, is it so that my splits of clusters is more due to quality or batches than due to actual cell types, whatever I'm trying to cluster? And thinking about this also with your clusters, how many cells do you need to find a cluster? If you have a rare cell type in your sample that is only, let's say, 2%, as in this example, of your cells, and I would require at least 5 cells, it will calculate here how many cells I need to sequence to get to this number. So it's really good to think about these things before you design an experiment. And 5 cells, I would say, is way too little to actually form its own cluster with these methods that we're using. So there needs to be a significant signal of this group of cells for them to stand out and cluster on their own. So 5 cells after integration, that might work, that all these 5 cells in 10 sample each would form its own cluster. But consider probably like maybe you need 50 cells of each cell type total from all your samples to even see it as a cluster. And then to do differential expression, you probably need even more cells to get good statistics on all genes. And also something that's really good to consider is to do subclustering. Once you have all your cells together from a tissue and you've done your clustering, it's a good idea to take one subtype of cells and rerun all the steps we've been doing. Because when we take all data, we select variable genes, we run PCA. Most of the variation in the genes and the PCA will be the difference between the main cell types, and will not describe so much the difference within this cell type. So if we then take only this cell type and we rerun all the steps that we've done before, we will get better separation of the data within that cell type. And as I said, always do your clustering. We talked about looking at QC on the UMAP, but also look at QC on your clusters. You might see something like this. Like here I have one cluster with really low proportion protein coding. There is something fishy with this cluster. Maybe I need to go back and look at my QC and filter it out. Or maybe we have a few clusters here with high percent mitochondria. Does it make sense? Is it cell types that are known to have more mitochondria, or is this also a technical artifact? So always after doing clustering, do these violin plots with your different QC stats, see if it agrees with what you believe, or is it some technical artifact that is causing these clusters to form? Yeah, more examples what you should look for. Sometimes it could make sense that you have differences in quality due to it being different cell types. Sometimes it will be technical artifacts, and to know which is super hard. So it's a combination of also a bit of experience that you know that which cell types are smaller, which are bigger, which you expect to have more mitochondria, less mitochondria, really knowing the biology of these cells and what might make sense. Also check batches and conditions. Do we have equal representation of our different samples in the different clusters? Is it making sense that we have like this one cluster that's mainly dominated by one sample? That could be a good thing that it's a unique cluster, or it could be that our integration failed completely.
I wanted to ask, in cases like this, do we try to reintegrate in another method or maybe 2 methods? And then if we see the same pattern, do we just exclude this sample?
Oh, I can't give one straight answer to that. I mean, it clearly depends. If that sample happens to have a bit like this cell type that, for instance, let's say that you have these biopsies that you are studying, and then you happen to have one biopsy that also got a bit of another tissue type into the biopsy. So that one sample will have a different cell type. Then I would exclude that cluster and motivate why I'm doing it in my method section of the paper. But if it's like that, all the cells of that sample stands out on its own in its own cluster and it's behaving weird, then yes, I would consider removing it and then motivate that in the method section. So there's not one straight answer, of course. You have to, it's always a bit of exploring, going back, looking at like marker genes for the clusters. Can I make sense of why only this sample has this cell type? Or, yeah.
But my question is more like, sure, even if if the difference is not biological, then like if it's some technical artifact or something like that, then I understand. I mean, I think removing it would be good, but then even if it's biological, my question is like, what kind of information would we get from this specific tissue that we've like extracted from a person, where we can see that the population is mainly comprised out of one cell type?
No, I mean, that clearly depends on how you got the samples, right?
Yeah.
So it's hard to say something generalizable, but it's really, when you have problematic samples, I can tell you that it's hours and hours and days working on like a detective to figure out. You look at all kinds of QC stats. You look at genes, you look at ambient RNA, background signal, doublets, filtering in different ways, trying to figure out what's wrong. Can I use this sample? If you have hundreds of samples, then you can afford to just throw these troublesome samples out. Yeah. But quite often you end up in studies where you have your 4 patients and your 4 controls, and then you don't want to throw out the one sample.
You're underpowered in a way to start off with.
Yeah, and then, I mean, I've had so many projects spending hours and hours trying to figure out these things.
That's fair.
That's
Thank
the
you.
fact of life. Yes, I'm back to the slides that I started with. What sort of assumptions you make in the beginning will lead to different results. So be a bit lax in making too many assumptions. Also let the data guide you and see what clusters you get and explore it. There's a danger in deciding before that we want 4 clusters and then tweaking the parameters to get 4 clusters and maybe missing out on important novel information that you didn't expect. And then try a few different methods, see if it looks the same. So to wrap up, in most cases, you will get the exact same clusters regardless of how you do it. It's often a good idea to take out your cells and do subclustering in a separate round if you have one cell type that's super interesting. Quite often you also need to rerun the integration then. So running several methods, having agreement also with the UMAP, that gives you confidence to believe that like the clustering is making sense, but it's really like looking at genes, using your biological knowledge to evaluate that is how to know what's good or not. So that's it. Do we have any questions on clustering?
I was wondering, can you also cluster based on a gene set that you select, kind of? That you make gene set and then do clustering? And does that make sense in some cases?
It does make sense in some cases. There's always like, I would say I always prefer an unbiased selection where we go by variable or something, because there's always like the risk of, you can always fit your data to force it into something with selecting gene sets too much. But it could be the case, like for instance, you have bulk RNA-seq, you have differential expression that like these genes change with this treatment or something, and you're trying to force out what are the cell types that are changing with this treatment. It should be used with care. Really, this like doing a pre-selection of genes, because there is a risk that you bias your data, there is clearly a risk that you will lose information that you might discover otherwise also by selecting genes.
Okay.
But for sure, I mean, there's a lot of people that do it, and it does work.
Okay, great. Thanks.
I was questioning about the subclustering. Like,
Yeah.
at what point is you think, like, is there a metrics to evaluate that the subclustering is good? Like, this is if one cluster can be divided into two subclusters and no more.
Oh, you
something
mean
like
by
that.
subclustering like that? Now I understand what you mean.
Yeah.
Let's see if but I mean, we can take this, just this plot as an example, because of course, is it true that these are distinct clusters, or should all of this be one single cluster? Those kind of decisions is something you will always have to face. My best recommendation is really to do differential expression between these three clusters, see if they make sense or not, to decide if the splitting of these clusters is what you should do or not. Also look at QC stats if it's like mainly just that you're having the low quality cells of this cluster that are separating. Look at cell cycle, is it mainly like cell cycle signal, or the different batches separating them? So that's more or less what you're stuck with.
Related with this, so when you want to validate your clusters, do you look at the genes driving those clusters, or you do one extra step of differential expression analysis?
I mean, from this graph clustering, we can't really find the genes that are driving the clustering per se. That kind of information is lost in the whole method. But so to find what's driving the clustering, we need to go into differential expression. Did you?
Yeah, I don't know if I understand how do you get the genes in cluster 0 if I want to like evaluate the biology of that.
Yeah, then I would do differential expression of cluster 0 versus 8,
Okay.
0 versus 6, and then possibly 0 versus all the rest, to try to figure out what is the signal in 0, and does it make sense that this is its own cluster? Because it will quite often be the case that all these clusters here will have similar marker genes when we're comparing them versus the rest, right? So if we're just doing each versus rest, they might all have the same marker genes, more or less. But then if we start comparing them within each other, they might also have really interesting marker genes that make some biological sense, and they may not. And if they don't, I would say there's no point in splitting into more subclusters.
Okay, thank you.
I have a question.
Yes, go ahead.
Between the arguments like Leiden and Louvain, how do we make the choice? Because I know in Seurat the default had always been Louvain clusters, and then recently in the latest Seurat they implemented the option to put argument equal 4 to use Leiden clusters. And I've seen in almost all papers when they do these sort of clusterings, they always use Leiden. So yeah, we've just kind of in my group just accepted that, okay, we're gonna use Leiden, but this was integrated in Seurat so recently that before it was a bit hard to do it. So how do we decide whether one is better?
Yeah, unfortunately, this is something that hasn't been like super much evaluated. There was some papers showing that Leiden was better, but not really that thoroughly compared, I would say. It's also, I think, a trade-off of just computational cost that is faster. In SCAMPy now, they're actually even just now implementing Leiden, because the Louvain Python package is not maintained by whoever developed it to update to later versions of NumPy and so on. So they don't keep it anymore. But I would say when I run both of them, there's they're not that different either. You get very similar results, so probably wouldn't make a huge difference to run either of them. So as long as Surat is also supporting Louvain, I think you're fine to run that also. It's not that big a deal.
Okay, thank you. And one other question, a bit specific to my work, because I've been struggling with this in the past few weeks actually, is that when I cluster a group of cells based on the cell type, so I have identified the cell types, and then when I cluster them, I was expecting to see like very individual or very marked different clusters, but then I always find some like very close or overlapping clusters. And I don't know if that is acceptable for me to present saying, I can assure that this cell type is not this other cell type, even though they are so close together in space and maybe overlapping a bit.
But how do you know what the cell, is it sorted cell types? Do you actually know what they are?
No, it's through a label transfer, like through
okay.
expression.
So then the question is, what do you trust more, your label transfer annotation or your clustering?
Oof. Yes.
Now I put you on the spot here. Yeah. We'll talk more about this, I think, also in the after cell type prediction, and in the end, what you can trust and using your common sense when it comes to these kind of things. None of the methods are perfect. So it could also be that your clustering is correct and your cell type annotation is wrong. Who knows?
Okay.
But we can look at it more also perhaps that we bring your own data day if you have your data.
Okay, nice. Thank you.
Yeah, I saw in one paper about cluster validity index. I mean, whether the cluster is good or bad, they are checking with different kinds of validity index. So those kind of methods can be applicable over here too.
But I'm not aware of this. Was this with single cell data or?
Okay. Yes, I didn't check it with single cell data. This is a normal clustering paper where they had used k-means, hierarchical, Lorentz, all methods. And after the clustering, they used cluster validity index like Silute index, Dune index. And they checked that which cluster result is good or bad, something like that.
So basically, it's sort of a consensus thing where they do multiple methods and give a score to cluster validity.
Yes.
I think that sounds like a really good idea. I haven't really seen it implemented for single cell data. If anyone has any links for it and want to share, would happily see something like that for clustering on single cell data. Would be nice.